For this last talk, we have Wani Wu, who's here from SKKU, Songkun Kwan University in South Korea, where he is a professor of biomedical engineering and the associate director of their neuroimaging research center.
Yeah.
Wani is known for his work on cognitive and affective neuroscience, and I think today he's going to be talking to us about using machine learning to identify biomarkers. So let's all welcome Wani.
Thank you so much. Yeah. It's great to be here. Thank you, Noah and Ariel, for having me. I always kind of enjoy being in summer school. I was in once the European pain summer school that kind of there I met so many good friends who are I'm still those are I'm still seeing in conferences. So you will meet each other in conferences in the future. Anyway, so today I will talk about good practices in developing neuroimaging biomarker. I will give you some big picture, not the really low level things. I mean, how to develop good models. I heard about the task you've been given, but I might not be giving you the ideas of how to build a model, but I will give you some big picture thing. I will start with some introduction that I'm in the Center for Neuroscience Imaging Research. That is a biggest imaging center in Korea, and I'm an associate director there. We have wait. Anyway, yeah, we have okay. We have 4 MRI machines, 2 for human and non-human primates, and 2 for rodents. So we have this great infrastructure for cross-species imaging. And this is my lab, computational cognitive affective neuroscience lab. We go by COCO1 lab. We are mainly using fMRI to study pain and emotion. And most of our lab members are Korean, but we are trying to increase diversity. So please, if you are interested in our lab, let me know. And I got my PhD with Torwager. He's currently at Dartmouth, and he got me into this neuroimaging biomarker research. And fortunately, the popularity of these studies kind of has been increasing. If we look at the PubMed, the number of publication on neuroimaging biomarker is been increasing. And last year, there were around 2,500 papers. And it's understandable because, the most this shows the 10 leading causes of disability worldwide and kind of top global burden. And 6 out of those 10 leading causes of disability are targets of neurotechnology, including depression, anxiety, and pain, headaches. All these should be benefited from non-invasive neuroimaging tools, right? And that can be combined with machine learning and AI techniques that we see the recent advances. And I study pain, and I will use pain as a main example today because I know that the best. And also, I think the neuroimaging biomarker studies is kind of well developed and kind of matured in this pain research area. And I often play this video kind of where Francis Collins, former NIH director, talk about the need for pain biomarker. I will play. It's a few seconds.
We need biomarkers that would be able to actually distinguish different kinds of pain in different populations, because the treatment for one kind of pain may be very different than another, and we do too much lumping right now of pain syndromes. And biomarkers that could actually predict clinical outcomes to a particular therapy. We need objective measurements of pain, what we would call a painometer, instead of the very long in place but not all that useful 1 to 10 scale.
There will be many reasons why we need this type of biomarker, but one of the most important reasons that I think is this one. We are now combat the battle against the kind of opioid pandemic, right? And then, so we need to develop new treatment for pain, but this strong placebo effects really thwarts painkiller trials. And so the NIH kind of HEAL initiative, actually, they made a program for developing these surrogate markers that is less vulnerable to placebo effect. That could be the brain, right? But if we look at the brain, pain in the brain, we really need a systems level neuroimaging methods because uh, pain processing in the brain is well distributed across multiple areas. So because I mean it's so understandable because pain involves many different processes like emotion and motor control, attention and feature extraction and so on. And even from the spinal cord, it's well known that pain processing has a degenerate nature, meaning that there are multiple structures and processes that can generate the equivalent functional outputs. That is a pain. And yeah, from the spinal cord, there are multiple pathways. So we need a systems level neuroimaging methods, and it is going to be the case for other leading causes of global burden as well, right? And we know the power of fMRI, right? So this shows the ongoing responses to noxious heat, and I mean, from reconstructed from our data, our lab's data, and also it provides high dimensional data that we can benefit from recent advances in AI. But more importantly, it really provides a platform for multi-species neuroimaging that can bridge between preclinical and clinical studies. So in the lab, we are now actually doing some research on rodents and also non-human primates and now human. I mean, I've been kind of trained to do fMRI study on human, but we are now expanding our area so that we can model these functional outcomes relevant to clinical conditions, and then that will provide a pathway to clinical translation. And one thing that I mean, one kind of big observation that I did, or what something that I learned from my training, the biggest shift was this, the predictive modeling approach. That is actually a little bit different from traditional mapping approach. I mean, you learned this in this morning by Elizabeth, but I will briefly touch on what's the difference. So in traditional mapping approach, we usually do kind of have participants do some tasks in the scanner and then ask this question, is there an effect at each voxel or region? So it's all about local mapping, right? There is no model for distributed information. Therefore, we kind of only can obtain observe the small effect sizes, and it requires massive multiple comparisons, and therefore we kind of lose a lot of power, statistical power. But predictive modeling approach really is about the reversing the equation. So now brain becomes an independent variable, and outcome becomes a y variable, like the dependent variable, so that the question becomes like this: can we model the joint contributions of multiple brain regions to outcomes? And uh, by relying on this brain-wide pattern-based information, we can actually achieve large effect sizes, and there's no need for multiple comparisons, and we can achieve high power, statistical power. And also we can benefit from this fine-grained mapping. You can filter indirect connections so that we can enhance specificity of identifying the connection between one brain region and the outcome. And we can also identify some mask effects that is enhancing sensitivity. I mean, I've been kind of arguing the benefit of doing using this predictive modeling approach in multiple publications around, I mean, when I kind of around my graduation, including this paper. So you can check it out. But we can also view this as uh, this predictive modeling approach from a different perspective as a way of establishing tight association between mind and brain measures. That is our goal. I mean, the goal of cognitive neuroscientists, right? I mean, there are kind of many cognitive neuroscientists actually do only this, like the mapping, the traditional mapping really map some functions onto the brain. We call that forward inference, right? In a probability term, that is a probability of observing this brain measure given some function, right? I use pain as an example. But even though many people stop there, we need to establish really large, I mean, examine whether that connection has a large effect size, right, between these two function and brain measure. And we can, I mean, we should not stop there though. The more importantly, we need to kind of establish this specificity. That is the probability of not observing that brain measure given there is no function. So there is a not pain and then no brain activation. I mean, there's a famous story about ACC, anterior cingulate cortex. You always see the activation in ACC, and that has really low specificity. So based on all these criteria, we can actually establish this positive predictive value. I mean, you heard about this from Ross's talk a little bit. And we call that brain decoding, and sometimes we call that reverse inference. That is the in a probability term, that is a probability of a function given our observation of that brain measure. So this is really the D is a diagnostic value of a brain measure, and we really want to establish this. And then kind of meeting all these criteria, that requires to say we established the association between mind and brain measure. And there has been many discussions about this inference and logic of these inferences. And I really recommend Ross's talk on this topic. Brain decoding, can fMRI read minds? Where Ross really explained well about the forward logic and backward logic and so on. Anyway, and kind of we did, like 5 years ago, we did a survey on studies that developed neuroimaging-based pain biomarker published between 2009 and 2019. And this shows the distribution of the neuroimaging tools over that number of studies. And you can see many people use the fMRI as a tool. It's understandable because pain is, I mean, the deep brain structures are really important for decoding pain. So fMRI is a good kind of neuroimaging tool. And then shows the study population, and majority of the studies really target the clinical population. And this shows the different tasks, targets, and model levels. And I mean, we did a kind of detailed survey. And one observation that struck me, that grabbed my attention, was the most popular approach was binary classification, like pain versus no pain, or case versus control, and population level model. That means that developing a model that works for everybody, and data level, one of the kind of popular, not the most popular approach, but individual level approach is kind of one of the largest kind of proportion. That means that we want to make inferences about each individual, and many studies really use the brain-wide pattern. So these characteristics really suggest that researchers who developed these models were mindful of the model's clinical applications. They want to use this model for clinical application. But we still don't see any good clinical application, right? Many years ago, like now, it's been almost 10 years, Peter Bandettini at NIH, he wrote a blog post. And in that post, he wrote this: we are just one clinical application away to see the second growth phase in fMRI research. And we don't yet see that one clinical application. We are still one clinical application away, right? What are major barriers? I want to talk about this, but before give you my take, I want to hear your thoughts. Yeah. I set up a slide yeah, and then can you take a minute to write your thoughts on what are the major barriers that kind of prevent us from seeing this clinical application of fMRI tools or neuroimaging tools? And I will give you my take, and then at the end of the talk, I want to kind of learn from you as well. Okay, I will give you a couple minutes. If you have any questions, let me know. You can interview me anytime. Okay, I will give you my take. I have three barriers in my list and one additional bonus. But anyway, so these three are the major barriers in my mind. And the first one is one publication and done attitude or approach. So this is another survey results that I did 10 years ago. I kind of surveyed machine learning models in clinical studies. And as you can see, maybe this list of clinical conditions include, your target as well. But at first glance, there seem to be many good performing models, right? Uh, it's um, the classification accuracy range from like 80% to 90%. It seems, they are good models. But if we put those in this biomarker development framework that has multiple stages, starting from development phase and prospective validation phase and robust replication phase and population level kind of deployment phase, we kind of thought that most promising models should be carried forward, right? And the later stages requires increasing levels of evidence. If we put all these models, the surveyed models in this framework, you can see most studies were stuck at the first step. Like majority of models just were at development stage. That means that people don't forward, carry forward their models and for prospective tests. You might think that sample size is going to be the problem, and actually we looked at divided the models into single site, multi-site, and consortium-based multi-site studies. And multi-site and consortium-based multi-site should have a larger sample size. But you might expect that these kind of models based on larger sample size have larger, higher kind of proportion for prospective testing. But it's a little bit larger, but not super higher. So you can see the green kind of studies like here, no prospective testing says many people just satisfied with one publication and stop there. And if we look at the model performance in prospective testing, it dropped kind of significantly. So the problem is, even with cross-validation, there is a bias in the report because of publication pressure and selective kind of report, right? And one interesting thing was observation was promising models had their names. So this is a table that we kind of compiled 10 years ago, kind of compiled the promising candidate markers. And, these models had names. And that, I think, kind of convey the researchers' intention that we will retest these models again for prospective testing. And when I shared this idea to my colleagues at the time, one person who has a background in education and computer science told me that, okay, those are boundary objects. In their field, in sociology field, they call that as a boundary object. What is a boundary object? That is an object, something that can be used in different ways by different groups, like specimens or field notes and maps. So boundary objects are plastic and can be interpreted differently across communities, but with enough immutable content to maintain its integrity. So after that conversation, I kind of started to view the brain maps or models as something that kind of tangible, that can be kind of accessible by multiple groups. So brain models can be used by clinicians, cognitive neuroscientists, or computer scientists kind of for different purposes, but brain map has the kind of enough immutable content, to maintain its integrity. So, there are platforms that we can share these models. We talked a lot about data sharing, but model sharing is another really important thing we need to think about. And I came across this Twitter post actually advertising Ross Poldrake's talk many years ago. But the title is like Scientific Publication Beyond the Text, Sharing Research Objects. I think sharing research objects is so important, and the science of neuroimaging can be more than publication. And one thing that kind of was shocking to me was, we published um, the neurologic pain signature back in the day, and then that was patented. Do you know the brain models can be patented? So it's been patented and to kind of protect researchers from lucrative kind of activities. Now, with this patent, kind of we share the NPS with a minimal kind of agreement with researchers. Yeah, so brain map. This is a new way of kind of viewing brain map, I think. That was kind of biggest kind of paradigm shift for me during my PhD. But there is one challenge in sharing models. That is a problem known as a toothbrush problem. No self-respecting person wants to use anyone else's. So we many times we don't want to use other teams' models on our data or vice versa. And there is another kind of challenge that is a kind of fear of falsification. If we develop a model and then we named it, and then we now got for example, some agreement, and then we sent the file. We do that in the lab many times, and then they kind of test our model on their data set, and then kind of send an email to us like, oh, your model doesn't work for our data. That kind of, yeah, that happens. And there is actually, I call that as a healthy fear, right, for researchers. We can be falsified, right? Our models can be kind of falsified. And I think that's a really healthy kind of fear. So yeah, the first barrier for me is, in my kind of thinking, is one publication and done approach. And the solution for that is to treat neuroimaging models as research objects that can be shared. And second one is focusing on easy problems. What I mean by easy problem is in terms of clinical utility. This shows the survey results, yeah, also 10 years ago, kind of showing the proportions of distribution of the clinical conditions, and the plot on the right shows the use of machine learning models in these studies. And you can notice that majority of studies really focus on diagnosis. It's because, there could be many reasons, but it's mainly because it's easiest problem, I think. And many data set has a clinical condition as a outcome variable. So it's kind of simplest one is going to be like case control classification. But whenever I talk to physicians, I mean, I have because our lab was kind of working on pain research, there were opportunities to meet physicians, and they always tell me that these models is not going to be kind of useful for them because they have a way to diagnose people. So the physicians told me that these 25% will be more useful, but these are much more challenging. And I came across this machine automation map once. This also kind of many years ago from Harvard Business Review. Uh, the machine automation map has two dimensions. One is risk and another is accuracy. And for example, credit card fraud detection, that was already automated because the accuracy was mediocre, but the risk is also really low. So the things that under this line is gonna, was kind of already automated. But for example, driverless car, I mean, it's now kind of being deployed, but it has a much higher accuracy than credit card fraud detection, but the risk is also really high, right? So that's kind of preventing from machine automation. And I thought there could be another dimension for clinical translation, that is clinical utility. For example, the diagnostic test, have been able to achieve high accuracy, right? So that means, like high accuracy, but the clinical utility is low. So because of that, maybe that is not going to be translated to the clinic that fast. But for example, early detection or risk assessment or monitoring biomarkers, those may show a lower accuracy than diagnostic tests. But because of their high clinical utility, they might be able to be translated into clinic earlier than diagnostic test. So kind of we argue that suggest that we need to concentrate our efforts on neuroimaging models with high clinical utility, including kind of these targets. And after that, I came across also this table that shows there are many different types of biomarkers. This is kind of something that produced by FDA and NIH Biomarker Working Group, and we don't know which biomarker will be more useful for which clinical conditions. And this paper, re-engineering neurotechnology, placing patient first, says something called 3S principle. And one of the S is stakeholders, including patients, caregivers, and healthcare providers. And this paper really argued that we need to involve them in the process early as possible, so that we first identify the problem, the target that is going to be useful for them, and then put our efforts for that goal. So I think in the big picture, we need to be really selective and informed by these stakeholders. So the second problem, the solution is going to be prioritize our effort to develop models with high clinical utility. And third one is prediction over understanding. I mean, I've been in this pain biomarker development kind of research area for now like 15 years. And then I got some nasty reviews sometimes, and this was one of them. The paper is very impressive regarding the massive computational technology that the author employed. I mean, I kind of used a linear model. So for them, there was like massive, computational technology. Anyway, however, the technology seems to have convinced the authors that they have discovered a new fundamental brain structural organization rule, where I think they are being misled by statistical tricks. I mean, as a pain researcher, we get kind of people who has a physiology background really often. And I think this review was made by some physiology researchers. Yeah. So, but this led us to do this project. And I mean, this paper, we wrote the paper focusing on the interpretability of predictive models. And interpretation of these models is a key to kind of communicate with those people. I mean, as a kind of technology savvy people, I mean, here you are really the one side, right? And but we need to acknowledge that the models are so complicated. For linear model, I mean, has that number of features is also like really large, and interpreting that even linear model is really complicated, right? But if we do not know why and how a model works, we cannot answer the following questions. When will the model fail? And to which individual or subgroups does it apply? And how can it advance our understanding of neurobiological mechanisms? And so in that paper, we proposed that to be interpretable, the model should be readable and understandable to human researchers, and provide useful information about neural representation and mechanisms of mental construct, and behaviors. And the model should show the predictions are based on true signal, not noise. And so we proposed three different levels of approach to the model interpretation. One is feature level, that is like lowest level, which feature is important contributors to the prediction. And the second level is model level, analyzing model behavior. Is that confound free, or is that vulnerable to bias, or does that has any weak points, whether that is a generalizable? And most importantly and most challenging step is biology level assessment, that we kind of seek looking for converging evidence from literature and more invasive studies. And if there is no kind of converging evidence, that may lead us to a novel discovery, right? Through the theory development and new experiments. Yeah. So we need these all three levels of interpretation, and we compiled all these different methods that are going to be helpful for these three levels of assessment. And also we proposed a protocol for an example workflow. And yeah, so step 1 to step 15 or something. And all codes are, yeah, written in MATLAB, sorry. It's not in Python. But but it could be useful. I mean, hopefully. Because I mean, part of the reason is our lab is still using Tor's catnap tools that is kind of massively written in MATLAB, and that is a great tool actually that has multiple toolboxes, and also kind of there are kind of good functions for kind of this machine learning kind of training, model training and interpretation. So we are still using that. Anyway, yeah, and also I wanted to point out that this component process approach might be helpful. What I mean by component process approach is, I mean, I will contrast that with a direct prediction approach. In the direct prediction approach, researchers use brain directly predict the clinical outcome. But in component process approach, we can establish some intermediate layer that is component processes that is closely related to the brain mechanisms, and then use these subcomponent markers to predict really complicated clinical outcomes. So yeah, we use this analogy. This is grayscale, and this maybe could be seen as a RGB scale, so that go with this multi-component processes. And what I wanted to say is this step, this level is what clinicians are interested in, but what neuroscientists are interested in is this kind of step. And by using this approach, we can actually integrate both groups' interest and maybe hopefully to bridge the gap between neuroscientists and clinicians. Uh, yeah, sometimes the translational gap is called the valley of death, and yeah, so we really want to close the gap. Okay. The solution for the third one is make advances in neuroscientific understanding. I think that's super important to kind of get the energy for this research. But are these all? And as time goes on, I've been in this field many years, and then I keep realizing that the real issue is something else. That is a poor prediction performance. That is really that bothers me like long time. And you saw this paper multiple times now, right? The Mark paper. Yeah, if someone asked about how many, I mean, how many samples you need to predict something, and then yeah, that requires thousands of individuals. Basically, that is the answer. But and this is a figure 4F of the paper, and that kind of suggests that there is a relationship between multivariate decoding and univariate decoding. But the real message, real point of this plot for me was the range of each axis. So the univariate models, we can achieve like correlation 0.1, less than 0.1. So each dot is a phenotypes. Something, I mean, uh, yes, including like intelligent IQ and age maybe. So I mean, some phenotypes are easy to predict actually, but with the univariate models, you can achieve higher. You cannot achieve higher than correlation 0.1. And for if you use multivariate models, yeah, you can achieve like up to 0.4, but it's still low. If you convert that into R square, I mean, we cannot just square the correlation, but I mean, if we do, even if we do, it's around 15% of explained variance. So it's still very low. And even kind of more kind of frustrating message was from 2022 paper where Bob Cogwell group, they show that we cannot predict individual differences in pain sensitivity. This is a main figure. The empty brain is a main figure. You know, the just brain template can goes to the main figure. That's a figure 2C that's showing that there's no region is correlated, no region that is correlated with the pain sensitivity across people. And these are the supplementary figures where they show that even with the multivariate models, they cannot predict pain sensitivity across people. Yeah, so this is a real challenge for us, and but this led me to think about doing this personalized mapping approach. And I went back to these types of biomarkers, and you know, if we divide, if we see the types of biomarkers, there are some types that is targeting within individual changes. So yeah, I thought our lab can target this first, and that's related to this. Yeah, the detecting the treatment effects is kind of detecting within individual changes. So we started this project, My Pain Connectome project, inspired by my Connectome project. You know, you heard about this, right? Ross scanned himself more than 100 times. And NPC is secretly also kind of parody of the midnight scan club, Midnight Pain Club, so MPC project. Yeah, but our goal was different. We conducted N=1 pain fMRI study to examine the upper ceiling of the accuracy of fMRI-based prediction of pain. So we wanted to know what is the maximum accuracy we can get from the fMRI, and to disentangle population versus individual level brain representations of pain. And this project was spearheaded by Young-eun, who is sitting here, during her master's. This MPC project has two different data sets. One is MPC1 that involves extensive sampling over one participant. So we did multiple scans for one participant, and MPC100 is another data set that is we scan many participants, but single scan. And N=1 participant was actually me. So we call the data as MPC Wani as well. At the time, when we applied for the IRB didn't approve to scan students. So because of the potential coercion, so they only approved scan the PI. So I entered the scanner more than 30 times. Each session was 2 hours, and I got two types of pain. One hip pain on the arm, and then capsaicin pain. And there were 5 sleeping sessions. I mean, intentionally slept through the session and didn't rate the pain. But anyway, I will show you some results of this 28 awake session where I got these two types of pain, and I will show you the results from hip pain stimulation. And usually we give the rating for each trial. So, and the goal was to predict trial by trial pain rating. And the second type, the cross-sectional study, was we scanned 124 people, the same design, but kind of one session. Okay. And yeah, as I said, the goal was to predict trial level pain ratings. So there was each trial, there was one hit stimulation and then rating, and then we scan the brain. So it's perfect for kind of this type of machine learning prediction. And we divided the data into training and validation and testing data. I mean, this is a really good practice. So that you use the cross validation to get the estimated generalizability, the performance, and then you can use this validation set for hyper-tuning of hyperparameters and fine-tuning of hyperparameters, and then freeze the model and then test on the unseen data, right? And we use these 3 types of modeling inputs. One is MPC 100 model, that is population level model. So the model developed based on this MPC 100. So kind of using the other people's brain to predict my own pain rating. And the second one was MPC1 model, that is personalized brain model. So using my own brain data to predict my own pain. And the third one was quite interesting. We used these experimental parameters, including stimulus intensity, skin size, trial orders. I mean, the pain rating will be largely driven by stimulus intensity, right? The temperature. And so, yeah, we wanted to see how the brain fMRI data can do above and beyond experimental parameters. And the results that I'm going to show you is the test results on this independent test data of MPC1. So the results can be summarized with this Venn diagram. The total R squared for modeling trial by trial pain rating was 0.684. It's large, right? It's kind of, I mean, and only with the brain, it was like 60% explained variance. This was the highest number that I have seen in entire my career. So I was so happy. And if we divide it into personalized model and population level model, only with the personalized brain model, we were able to explain 60% variation of trial by trial pain rating. And population level model, with the population level model, we were able to explain 26%. And I was not particularly surprised by this number because one of my previous study, explained similar amount of variance using the population level model. So I can say the ballpark of the explained variance that we can achieve with the population level model will be for trial by trial pain rating will be that number. And interestingly, if we use these experimental parameters, the R square was 0.535, and the personalized model unique R square was 0.124. So you can see this is the unique variance explained by personalized model above and beyond the experimental parameters like stimulus intensity. Yeah, so the main point, 2 points are with the personalized brain model alone, we were able to explain around 60% trial by trial variance in pain ratings, and personalized brain model can provide prediction above and beyond the experimental parameters. And that led us to draw this figure. The question is, what is the variance that can be captured only by the brain? Because the observation was the personalized brain model can provide prediction above and beyond experimental parameters. That is external factors, right? And we usually think that this correlation as a brain behavior correlation. And the fact is actually brain decoding, brain-based biomarker can capture more variance than these behaviors driven by external factors. And yeah, the question is, what are these internal factors that can be reliably captured only by the personalized brain model? Yeah.
How about in rating? Is this a rating that you were putting yourself, or is this a rating that was used to generate the pain?
No, so after I get pain stimuli, the heat stimuli, I rate how much was that painful? How much painful was that, right? After the heat stimulation. So that was the rating. So each continuous. Yeah, it's kind of, we call generalized labeled magnitude scale, but it's continuous. It's not kind of same interval, but anyway, so it's continuous, similar to visual analog scale. So each trial has the stimulus intensity. We had 6 levels of heat stimulation and continuous rating. So rating is a subjective kind of pain intensity. Okay? Yeah. And yeah, so the question is, what are these internal factors that can be captured by this personalized brain model? And I went back to old literature. This figure is drawn by Ronald Melzack, who is a big figure in pain research, actually based on Montreal. I mean, he passed away a few years ago, but anyway, this plot shows the one arrow, outward arrow, that is a pain perception. But there are many inward arrows, and only one of them is phasic noxious input from the skin or sensory input. And the other inputs are all really internal. For example, one is basic input from the brain, attention, anxiety, expectation, and intrinsic neural inhibitory modulation. And this is interesting, tonic somatic input. Even if you are getting heat stimulation in the scanner, you are getting other tonic somatic inputs as well, like from trigger points and other parts of the brain and body. And visceral inputs, like inputs from the internal organs, and tonic inputs from the brain. That is a cultural learning, past experience, personality, and so on. And there are many autonomic inputs. All these inputs are always there, and our pain experience is kind of influenced by those as well. And our brain marker, brain activation is reflecting those variables as well. So I can say these are the sources of reduced reliability of behavioral measures. So even though you are getting the same level of stimulus input, you will feel different kind of levels of pain intensity. So that is a source for the reduced reliability, but some can be captured by the brain. So brain-based decoding, brain-based biomarker can actually explain more than maybe external kind of inputs. Yes, this was one, yeah.
So, well, this is very informative. I don't think most of these can be reliably captured while doing the scanner. Would you allude to that?
I mean, yeah, it's not, we haven't shown that for sure. I mean, empirically, but I mean, I'm kind of my kind of guess is, I show that the brain-based biomarker can explain unique kind of variance of like 12% above and beyond the external input, right? That might be those. And then I mean, the future study should show that these variants that only captured by this brain model are those. But I mean, yeah, so it's kind of my guess, right? And kind of gave us the future direction of the research, right? Yeah. What are the variants that is captured by personalized brain markers? I think those internal factors could be one of them. Okay. And how much data do we need? If we use the whole data, like that is 18 sessions of training set, we were able to explain 61% of variance. And if we use just one session data, 90 trials, for example, we can explain 17%. So it's kind of low, much lower than that. But as you can see, the rate of increase is kind of slow down over the increasing data size. So maybe there could be some sweet spot that maybe like 7 sessions, like 600 trials. With that, we can explain 50% of variance of trial by trial pain rating. So yeah, we now push our lab kind of hard to to this direction of like personalized predictive modeling approach. But then what about individual differences? As I showed you, the Hopefully et al. Showed that empty brain and kind of show no results. And actually, we did the same analysis with them with the MPC 100 data set, given that we have this 124 data points sample size. And then we actually found some significant results. Yeah, some brain regions were correlated with the pain sensitivity. And also we were able to predict, those individual differences in pain sensitivity with the MPS. And also when we apply the lasso PCR, we were able to predict, we were able to get I mean, not super high, but significant results. But why? Why they got no results and we got significant results? Our guess is a sample heterogeneity. I mean, this is another guess. I don't have a strong kind of evidence, but based on the age range, the Hopefully et al. Their data intentionally, includes kind of the data from larger age range, so and from community sample. But ours has lower age range, and also our sample kind of largely were undergrads and graduate students. So, and I thought this could be right because I mean, at the kind of same time, I mean, around that time, this paper came out. I mean, there was a Marek paper, and then there was Matters Arising paper for that paper, and then this paper is their reply to metals arising piece. And then they say, yeah, HCP is more homogeneous sample, and then ABCD is a more heterogeneous sample. And actually, they show that the out of sample correlation, that is a performance, is larger for more homogeneous sample. But a larger sample size is always helpful for poor generalization when the sample is more heterogeneous. But I mean, these two messages kind of, if you think through these two statements, they somewhat contradicting with each other, because larger sample size means that the sample could be more heterogeneous. And that led me to think that maybe these two statements has the implications for maybe different data sets. The first one has a implication for test set, and the second one has an implication for training set. I will give you an example. So we actually use this MPC 100 data as a benchmark data, and to test that idea, we average the data. Average the data over multiple trials, separately for training set and test set. So there are multiple plots in the paper, but I will show you just a couple of plots. So yeah, in this plot, the x-axis is the test data level. That means that there is this is no average over trials for test data. And if you go to right, there's a more averaging of test data. So this is averaging data over 16 trials. So that means that lower variance, right? We are reducing variance by averaging. So lower variance over averaging. And y-axis is a binary classification. That was like high versus low pain classification. And as you can see, the accuracy is increasing, right? Over kind of the reduced variance in test data. And I mean, in the morning, you kind of hear that Elizabeth said that if your test set has lower sample, the variance is increasing, right? So you can see the variance is increasing of the performance, but but still the accuracy itself is increasing. But there is another point that this line is actually when we average the train set over 16 trials. And if we do not average the train set, actually there is a jump in accuracy. So that means that higher variance in train set is actually helpful for improving prediction accuracy. And it was same for regression problem. This is a kind of rating regression prediction, rating prediction. So yeah, the same trend were observed. So the point is lower variance in the test set could enhance prediction performance, whereas greater variance in training set can improve the model generalizability. Yeah.
But that lower predictive power of the data sets then, if you need a heterogeneous test data set that high accuracy.
So can you repeat?
If having a heterogeneous test set results in lower predictive performance, won't that mean that if you have another unrelated data set, it would also have low performance for predictive power?
Yeah, so if the issue is you have to have so if the test set is kind of out of distribution, right? Your training set, your performance will become really low, right? So yeah, so that means that if the test data is heterogeneous, then generalizability will become lower.
Yeah.
That's what you asked.
If
Okay, I mean, this is, I'll keep going. Yeah, this is consistent with this paper, the main message of this, another Nature paper by Todd Constable group. They say, yeah, this is a quote from the paper. Model reflect stereotypical profiles. So model also contains some information about stereotypical profiles of training set, and models fail when applied to individuals who defy them. So model failure is reliable, phenotype specific, and generalizable across data sets. That means that models actually reflect kind of the patterns within the training set. So if you want to develop more generalizable model, your training data has to be heterogeneous, like has cover the larger variance, right? The distribution. And one, I mean, so Abigail Green, who is the first author of the paper, actually posted multiple Twitter posts at the time. And this third message was kind of quite interesting to me. Yeah, models systematically fail because they aren't predicting unitary cognitive constructs. They are predicting measures of these constructs intertwined with socio-demographic and clinical covariates, that is, stereotypes. So one important point is that what a predictive model captures may differ from what we assume it captures. So it's kind of the constructs, they are not one thing that is intertwined with other things. And I think that really kind of is consistent with the message that actually, yeah, Timothy Brown kind of stopped, I mean, a couple days ago. Yeah, so we need to establish heterogeneous sample to make a more generalizable model. And so it's the same for pain. Pain is not one thing. Pain is a really complicated, complex, and integrative brain state. So we really need this component process modeling approach, and I added multi-component. So yeah, social demographic could be one of them. And the discovering and developing good component targets, intermediate component targets, is going to be very important. And so in our field, in pain biomarker field, we are kind of moving slowly towards the this direction, developing composite biomarker. So not only using just brain measures, but we are also trying to incorporate many other modalities. So there could be, yeah, one direction. And in pain biomarker field, there have been many relevant issues that I didn't cover today, that I cannot cover today because of time. There have been multiple consensus statement for the kind of brain biomarkers, brain imaging tests for pain. And recently there was some comment going back and forth in Nature Neuroscience about what is the ethical issues. So because, yeah, so I think the field in pain biomarker, brain biomarker field is kind of becoming mature. And so we kind of wanted to write this paper that kind of summarize 15 years of neuroimaging based pain biomarkers. And that includes multiple issues. Stay tuned. We are writing this paper right now. And yeah, I want to end the talk with some recommendations on study design for biomarker development. And I mean, there are a lot, but you can read it later. I put that in PDF. And yeah, I want to look at the Slido results, but yeah, so I will show you maybe. Okay, how can I? Okay. And if you have any comments or questions, you can also put that on the Slido. Group average approach instead of precision and individual. Yeah, so this is, I think, yeah. we need to maybe, I think, you know, the important point is we need to know what fMRI data is kind of strong for, or if you are working with a structural MRI, you need to know your data, I mean, what your data is strong for. And then I think fMRI is kind of has a strength. It's really good for doing this precision or personalized mapping. But for capturing individual differences, I think fMRI has a weaknesses. Many articles are done for specific trait, non-generalizable and reliable for specific population. Yeah. Not enough and robust results. Yeah. It's related to performance and generalizability. So many there are so many barriers. Low signal to noise ratio maybe forces the push to large group studies. But we need personalized medicine approach most of them. Yeah, I agree. Humans' inability to handle 20T MRI. Yeah, I think, you know, the point of the Peter Bandettini's blog was about one possible solution could be the MR technology, the advances in MR technology, like real temperature, right? The MRI stuff. Educational clinical policies may pose a barrier to implementation. Yes. Cost benefit trade-offs. The field hasn't traditionally considered individual brain characteristics. Yeah. Lack of generalizability. Yeah. Individual differences. Yeah. This is like generic. I mean, we are different from each other. Yeah. Generalization, lack of knowledge on how to. Okay. Yeah, if you have any more idea, please post, but open for question.
There's a question in the Slack channel. So,
Okay.
I'll just read to you. What advice do you have about making sure that the models we develop are used appropriately by others?
Appropriately by others?
Yeah, so like corporations maybe, or
Cooperation. Yeah, collaboration, you mean, or the
no,
companies.
like if we develop a model and then another company needs to use it for prediction.
Oh, yeah. Yeah, that's a good question, actually. Yeah, when we developed this MPS, actually, one company who like that is we didn't know about the existence of the company, but they kind of advertise that they can read out the pain, and then they kind of put our marker, our paper as a advertisement. Yeah, so it was like really scary. I mean, so yeah. I think good practice will be, yeah, you can also apply for patent to prevent some lucrative kind of parties use your models kind of in an unwanted way. I mean, the way you don't want. And also we have this like, the agreement is kind of, I mean, in our lab, when we develop good model, we don't share that kind of openly, but kind of we put that on the kind of, we put a little kind of, what is that, share layer of agreement kind of paperwork. So they sent me the agreement, kind of, what is it, the non-disclosure or kind of keeping the secret together, like mutual kind of agreement of that, keeping that, not sharing that openly. And then with that agreement, we send the model. So yeah, I think that really, at least with that, allow me to screen what people are using our model on which purpose. And yeah, so because of the worry about these kind of companies, like they may make money out of your work. Yeah, unknowingly. Yeah.
So I have two questions. One is a more technical and another is a little bit more philosophical. So on the technical one, I wanted to know that we're saying that you're basically predicting this pain subjective pain ratings from brain. And I wonder if there was also an objective angle to it, because the pain stimulation thing that you had, I imagine there are some parameters that someone was setting maybe, that would cause the pain. And I wonder to what extent, like you've thought about the subjective versus objective component of the pain. And the second technical aspect was like, where are you decoding, or not decoding, but like predicting these pain ratings from, like what region were this like related to somatotrophy, which might explain such high R square, or I want to know where you were able to get such good
Where in the brain, you mean?
and the philosophical angle is about the clinical translation of fMRI-based measures. So one, I'm just wondering that when there is a pain in the body, that is because of some underlying issue, and that would need maybe physically addressing where the pain is coming from. So I'm wondering, like, what angle you were thinking about when you think about clinical translation of fMRI measures, because physical pains probably cannot be resolved with fMRI brain scanning. So I'm wondering.
Yeah. What is it, the end goal, and then relatedly, can we identify kind of the where in the body kind of is causing pain, right? I mean, yeah. The end goal, what is the end goal of doing this, right? I don't know. The first, I mean, as I said, the goal could be quite different across groups. So, I mean, if we make a model that can be used differently across, I mean, by different groups. So I kind of have a little bit flexible. I could be flexible for the use of, how that is can be used by many groups. But one thing that in my mind is, as I showed you, it can serve as a surrogate marker of kind of measuring the treatment effects. So we show that our neurologic pain signature, for example, that is not influenced by placebo effect. So having one brain marker that is not responding to placebo effect, I think that having those markers is really important to be used in clinical trials. So I think whenever I was asked which area could be the first one that your biomarker can help, is I think, yeah, the drug development and clinical trials. And the other area could be the treatment planning. Like, for example, we can use, I mean, we recently published a paper of the for decoding spontaneous pain of fibromyalgia patients. And that can serve as a monitoring biomarker, how much pain is this patient in right now. And then depending on but I mean, it's really hard to use fMRI to monitor the pain, ongoing pain. But anyway, but we can monitor those people and then kind of use that as a goal, I mean, as a way of planning treatment. And also, the other really big thing is to understand the mechanisms of pain. Yeah, so that is yeah, by kind of establishing this tight association between pain, for example, spontaneous pain and brain markers. I mean, we can kind of contribute to understanding of pain mechanisms in the brain. And yeah, the second question is the somatotopy, right?
Which region of the brain?
Which, yeah, I mean, it's distributed. And, one thing that we are keeping learning is actually, for example, chronic pain. I mean, in this thermal pain, somatomotor areas are important, for example, and insula and S2, there are kind of usual suspects of the pain predictive regions. But But for chronic pain condition, those regions are not that important. Actually, totally different set of brain regions are uh, contributing to, explaining each person's pain. So now we are seeing, so that recent paper about fibromyalgia patients' spontaneous pain. We scanned only two people, and two patients have totally different set of the brain regions, and they didn't cross predict the other's pain. So that means that, the mechanisms of pain for each person could be quite different from each other. So this type of understanding may contribute to the understanding of chronic pain. Yeah, but
Thank you for the great talk. I'm very interested in the third point under your barriers that prevent the clinical translation of MRI. Because you said it's because prediction over understanding, right? And it reminds me of Tony Coney's, who is also for this student, his paper called Choosing Prediction Over Explanation in Psychology. So he argues that psychology is itself is explanatory. And he argues that psychology should move toward being a more predictive science, which seems to contrast your argument.
That's a great question. Tal Yarkoni, he's a great thinker, and I was influenced by him a lot. We share some time in the lab together. But that paper was targeting psychologists, right? And I'm targeting the biomarker developers. Like most of the papers and models that I surveyed were from actually computer science background. For example, engineers, they just they kind of apply their algorithms without thinking, many times. So I'm saying that we need to focus on mechanisms, understanding. So I think the important thing is keeping balance between prediction and understanding, like pragmatic kind of goal and epistemic goal, and we need to keep balance. So I'm like in pain field, they treat me as a engineer, like engineering oriented person. I'm not. I try to convince them I'm also caring about understanding. So that's why I put those. And so there is a really two worlds, I think, two cultures, but we need to be in the middle, I think. Yeah. Thank you. Yeah.
I was wondering if the effect that's being studied is inherently small, would it increase by having large trials or subjects anyway? And then, would this be an issue where people just want to recruit more trials or something?
Oh, okay.
Could you repeat the question?
Yeah. So the question is, just increasing the number of data is gonna be solve the problem, right? So is that the question? So blindly increasing the number of, for example, data points from one person or across people, will that solve the problem, right? Maybe not. We know. I don't know. We need to increase data quality as well, and there are so many factors that we need to care at kind of simultaneously with increasing sample size, right? Yeah, like increasing signal to noise ratio. For example, in our lab, we recently tested multi-echo sequence, and that increased the prediction accuracy a lot, especially predicting individual differences. So so the higher prediction depends on these type of things like sequence and data quality, signal to noise ratio, And also, yeah, one thing that I recommended in this, yeah, here. So task design is also really important. Like maximize variance in both features and response for training set. I think, yeah, this is also important to kind of important considerations when you design the task. Yeah. I mean, so here, task design. So I think my point is the biomarker development started from task design. So data collection. So yeah, that's 2 cents from me. But more data is always helpful.
Thank you for the talk. It was really interesting. And on this point of the sample size, do you think that the sort of a normative modeling approach by which you try to learn a general structure of, let's say, pain response, and then you transfer to specific subjects,
Yeah.
would reduce the need for single subject data, or is it something that you have tried and you planning to try?
No, we haven't tried the normative modeling approach. And I think, yeah, I heard a lot about kind of promising results from normative kind of modeling approach. So yeah, I'm interested in doing that. Yeah, it's kind of, and I mean, while I'm listening to your question, I was also thinking about this maybe what is that, the foundation model, for example, like pre-training data and then fine-tune then model for each participant. That may also be a good idea. So yeah, normative sampling. I mean, so point is there could be other ways to maybe to solve the problem with a lower sample size, less sample size. Yeah. Okay.
Just typing online, but no question.
Let's see there. Cost. Okay. Funding expensive. Yeah. I will. Yeah. Sure. This.
There's so many comments on like the funding aspects, but I know FMI is not that expensive, not as expensive as PAT, actually. So
Oh, yeah.
the PAT is widely used and the.
Yeah. Exactly. Yeah.
So how do you think about like the multimodal imaging and the top?
Yeah. I mean, multimodal imaging will be the way to go. And then EEG is kind of really one of the kind of economic choice maybe. So in pain research group, yeah, there are good scientists who are using EEG to predict each person's pain. Yeah, so EEG could be used, and yeah. So PET, I haven't done any PET study, but so you are saying PET is more expensive than fMRI?
Way more expensive.
Maybe more. Okay. That's
For
kind of
example, a single tau
good to know.
scan would cost 4.5K. As opposed to about $700.
Yeah.
500 someplace for a
In
month.
Korea, our scan fee is really cheap. Like $200 per hour. So come to Korea and
Thanks.
use our scanner. And high quality.
Okay.
Okay, I think it's.
Great, well, let's thank Wani again for this great talk.
Thank you.
Here.